All right, so welcome back. This is going to be part 4 in our cells lecture. And here we're going to be looking at the process of protein synthesis. All right, so we've alluded to the idea of protein synthesis a few times now with this idea of we're going from DNA to RNA to protein and in what we call the central dogma of biology or the central dogma of genetics, right? So when we look at this process of protein synthesis, we first have to understand some terminology, all right? And so this slide here is showing you some of the terminology that's going to be discussed and going to be used as we talk about this process of protein synthesis, right? So one is going to be the chromosome, right? So we've all heard of chromosome. Chromosome is one form that our DNA can be in, or one of the formats of our DNA, right? It is when the DNA is highly condensed, and so it is very tightly packed and coiled together, and you kind of get that distinctive X-shaped structure that we think of as a chromosome, right? We also have something called chromatin, all right? And so chromatin is the relaxed form of a chromosome, right? So we have both our DNA, but it's just what is the form of that DNA? Okay, is it tightly compacted or is it more loose? Chromatin is more loosely arranged. All right. So we have this euchromatin, which anytime you see the term EU, that refers to true. So this is the true chromatin, loosely coiled around these blue histone proteins that you see here. And this is when it is open and ready for gene expression. When we have this interphase chromatin, this is more relaxed. And then here, this is when it is tightly coiled around DNA, which is heterochromatin. So this here is heterochromatin, tightly wrapped around these histones. This is euchromatin, when it's more loosely wrapped around and can actually be used for protein synthesis, right? And so this DNA in the form of chromatin in the form of chromosomes is located in the nucleus, right? So here's our cell. Within the cell, we have our nucleus. Within the nucleus, we have the chromatin, or it can condense into a chromosome, right? But you can see how both chromatin and chromosomes are just DNA. They're just DNA in a different style or a different format, okay? So the first phase of protein synthesis is going to be called transcription, all right? The second phase is going to be known as translation, okay? So transcription, if you think of what you do if you're transcribing something, right? So stop thinking in a biological sense for a second and just think in a general kind of everyday sense. Transcription is you are transcribing something, right? So if you're taking notes as you're watching the video, you may be transcribing what I'm saying, right? I'm speaking, right? So I'm speaking English, and then you write down what I say, you write it down in English, right? It's still the same language, it's just in a slightly different format, okay? Instead of spoken, it's written, right? So transcription, we're staying in the same language, okay? And this language is the language of nucleic acids, or the language of nucleotides, right? And we go from DNA to RNA, okay? So transcription, this is the big thing with transcription, you go from DNA to RNA. So we're making our RNA, okay? The second phase is called translation, all right? Translation, if you're translating something, right, you're going from one language to another, right? So if you're translating what I'm saying in English, you may be writing it or recording it in Spanish or German or French or some other language, right? So in this process of translation, now we're going from the language of nucleic acids to the language of proteins. Or if you're talking about the monomers, we're going from the language of nucleotides and we're going to the language of amino acids, okay? A very nice way to remember this in terms of which one comes first is to look at the words themselves. So they both start with trans, and then the next letter in both words is going to be C in transcription, and then the next letter in translation is going to be L. Well, C comes before L in the alphabet, and so C with transcription is going to occur first, and translation is going to occur second, right? Transcription will occur in the nucleus. Translation will occur out in the cytoplasm, all right? And you can see this schematic here to the right. Here is transcription taking place in the nucleus. Then we have translation occurring here out in the cytoplasm, all right? So keep these things in mind. Transcription, DNA to RNA. translation, RNA to protein, right? So that's the DNA to RNA to protein, which is the central dogma of biology, right? So when we start this process, right, when we start the process of making our RNA, we have something, or just in general, this is going to be referred to as gene expression, okay? So a gene in biology and genetics, right? A gene is just a segment of DNA that codes for a protein, right? The protein could be for hair color, it could be for eye color, it could be for a particular enzyme, right? It could be lots of different things, okay? So the first step is we have to start at something called a promoter region, okay? A promoter, right? The enzyme that's going to be involved in this step is called RNA polymerase, okay? And it's going to bind to a promoter on the DNA, okay? We can then begin at the promoter and we can go down our DNA, this RNA polymerase, and during this going down of DNA, we can produce RNA, right? So we can begin transcribing DNA into RNA, all right? And then at the end of this transcription, we're going to make something called pre-mRNA. And we'll talk about why it's called pre in a second, all right? Now we mentioned just now, this is the first allusion to mRNA, right? So there will be different types of RNA that we'll see, all right? One is called mRNA, one is called tRNA, and one is called rRNA, all right? So M stands for messenger. This is serving as the message to take to the ribosome to then be transcribed into a protein, all right? The second is called tRNA. This stands for transfer RNA, and we'll look at transfer RNA when we get into translation. And then the third one is rRNA, which stands for ribosomal RNA, and this is used to make up the ribosomes themselves that we'll look at. Okay, so mRNA, tRNA, and rRNA. The two we'll talk about most will be the mRNA and the tRNA. Okay, so transcription factors are going to generate a pre-mRNA from DNA. So this is essentially saying we're going to have something bind into a receptor in the nucleus, okay? And this will be something you talk more about when you get to endocrine system and AMP2. But it may be a hormone or it may be some type of molecule that stimulates the process of protein synthesis, okay? This could be called a transcription factor because it stimulates transcription to begin, all right? And we make our pre-mRNA, and then the pre-mRNA has to be modified to go from pre-mRNA to just mRNA, right? And we'll look at that in just a second, okay? So whenever we have our pre-mRNA, right? So we've started this process of making the RNA, okay? Anywhere that we had an A on the DNA, what letter did we put down? What complementary base pair did we put down that we talked about in the last lecture? Well, if we had an A in DNA, you should know that we put down a U for RNA, right? Why do we put a U? Well, first off, we don't have T's in RNA. And second reason is because A's pair with U's in RNA, right? So A's and U's. If we had a G in the DNA, what did we put down? We put down a C in the RNA. If we had a T in the DNA, what did we put down for RNA? We put down an A, right? So we can still have A's in RNA, right? So we get this long strand of A's, U's, C's, and G's. And at a certain point, we get to a terminator region or a terminator sequence of the DNA, and we stop transcribing, okay? Once we stop transcribing, what we're left with is something called pre-mRNA. Okay, that this term pre refers to the fact that it is not finished yet. Okay, it's pre, it's a prerequisite to what we're going to get to in just a little bit, right? You can look at this segment of RNA, this pre-mRNA, and there are certain areas that are good, that are coding for a protein, and there are certain areas that are not coding for a protein. If it codes for a protein, that is called an exon. If it does not code for a protein, that is called an intron, okay? So if you know that there's areas of the pre-mRNA that code for a protein, and there's areas that don't code for a protein, you go in and you take those areas that do not code, you take those segments that do not code out. Okay. In other words, you splice them out. All right. And this is accomplished via the help of an enzyme called a spliceosome. Okay. So a spliceosome catalyzes the removal of introns from a segment of pre-mRNA. All right. So you can see here up on this upper region of this image to the right, you have yellow regions, which are introns, and you have green regions that are exons, right? Well, we said that the introns are non-coding, so we don't want those. The green are exons that are coding, so we do want those. So you can stabilize this pre-mRNA by adding some things to the end, and then you can take your spliceosome and splice out these yellow introns, right? So if you look at these products down here, you get 3 different products that do not have any of those yellow introns, right? Because we splice them out, right? However, you can also get something called alternative splicing, right? And alternative splicing is when you splice out different introns, but you may also take out certain exons, and thus you can get different protein or different final mRNA, and thus different proteins from the same pre-mRNA strand, right? So from this one pre-mRNA strand here, we ended up with 3 different and 3 separate finished mRNA strands. So that means from these 3 strands here at the bottom, we'll get 3 different proteins, okay? So it's one way to utilize and make the most of a particular segment of pre-mRNA, okay? So look at this sequence of letters down here. Look at the sequence of letters down here and tell me what is the word that is hidden in this sequence of letters, okay? So pause the video, maybe look at it, see if you can find the word that is hidden, okay? It may be kind of difficult, all right? So what this is demonstrating is this jumble of letters here is similar to this pre-mRNA strand over here. It's got letters that are important, it has letters that are not important, letters that contribute to whatever the secret word is, and letters that are just there as noise that do not contribute, okay? So what you can do is you can take your spliceosome and you cut out those unnecessary introns, or in our example here, you cut out the unnecessary letters, right? So say we cut out this letter, all right? So maybe after every time we cut out a letter, see if you can determine what the word is, all right? So we cut out that letter, and then we cut out this one, and then we cut out that letter, and then we cut out this one right here, all right? So what's the word now? Look at this now and tell me what the secret word is, right? Well, the secret word is cat in the hat, right? Because we spliced out all of those excess or all of those unnecessary letters. This is in principle the same thing we have happening to our pre-mRNA. We splice out the unnecessary segments of RNA, and what we're left with is the final polished mRNA strand that can then be used in translation. Okay, so hopefully this makes sense for you, right? You can also add some things to the end of the pre-mRNA strand to kind of stabilize it. This is called a poly-A tail and a 5' cap. This is essentially this poly-A tail. It's just a bunch of those A's, a bunch of those adenines that are added onto the end, and the 5' cap is just a cap that's similar. And these help to prevent the breakdown and the degradation of this mRNA before it actually gets used. Okay, so it's a protective mechanism. All right. So here's a video or an illustration. So that was all transcription, right? So we had RNA polymerase come in and polymerize RNA. So we take all of our letters. Everywhere in DNA where we had a C, we put a G. Everywhere we had a G, we put a C. If we had an A, we put a U. If we had a T, we put an A, right? We then had our splicing. We had our alternative splicing, right? And we got our finished we got our finished mRNA strand, okay? And all of that occurred in the nucleus, okay? So the next phase is translation. So now we're taking that mRNA and we're going to translate it into something new. In other words, we're going to translate it into a protein, all right? So what you have happen here, if you look at this schematic, you have all of these little kind of green structures here bringing in these circular components, all right? These circular components are the amino acids that are being brought to the ribosome, okay? So I'll play this again. So here you see the yellow ribosome and all these colorful dots. These are all the amino acids. And these kind of green structures that are fading off here, these are the tRNA molecules, okay? So what did we say T stands for? The T stands for transfer RNA, okay? So the tRNA molecules are transferring amino acids to the side of the ribosome to be assembled together to make up our protein, right? Because we said previously that the monomer of proteins are amino acids, okay? Now, you'll notice, and you can see these right here, that on every tRNA molecule, there is a set of three letters, okay? Okay, so as I was saying, these three letters here that you see on the bottom of the tRNA molecule, these are called anticodons, okay? So anticodons. Three letters that you see here on this mRNA strand, these are called codons, all right? And the anticodons and the codons are complementary to each other, meaning that they pair up using our base pairing rules, right? So A's with U's, C's with G's, U with A, G with C, right? So on and so forth. So this UUU anticodon on the tRNA will match up with this AAA of the codon, right? So this AAA codon on the mRNA strand, okay? This GGA codon here, or anticodon here, would match up with a CCU codon here. So here is a CCU codon right there, all right? So these are always going to be complementary. And remember, tRNA is still RNA, so you're still going to have U, and you're not going to have T. All right. So this is the kind of basic sequence of events that happens. You have the mRNA join with the ribosome out in the cytoplasm. Ribosome is going to move and it's going to shift and begin putting these amino acids together that are coming from the tRNA molecules. And the tRNA molecules are what actually bring the amino acid to the site of the ribosome to be assembled together. So you can see here, here's going to be one tRNA come in. It will have its amino acid bond to the next one. The tRNA will shift to this exit site, and then it will be dislodged out of the ribosome. So notice that these tRNA molecules here coming in have their amino acid. Here they lose their amino acid, and then as they leave, they do not have it, and they'll go out and they'll pick up a different or they'll pick up the same amino acid, but they'll pick up a different one because they left theirs off right here. Okay, and we'll see in a second how you can figure out what codon on the mRNA is going to code for what particular amino acid. Okay, that's called the genetic code. Okay, so here's kind of a better illustration of what's happening. Okay, so ribosomes are or tRNA molecules are bringing in our amino acids. So these circular structures up here are our amino acids. The tRNA molecules, if you notice, they have those three base pairs, so that anticodon, and they'll match up with a codon on the mRNA strand. Okay, you can think of the mRNA strand shifting, or you can think of the ribosome shifting, but it's going to move the tRNA molecules over, and then they will leave. They'll go off and they'll bind another amino acid that's the same as the one that they brought before. They'll come back to the ribosome, and then they're ready to be used for the next time they're needed, right? So the big thing here is keep in mind codons are on the mRNA, they're on the messenger RNA. Anticodons are on the tRNA, okay? And the codons and the anticodons are complementary, okay? Each of them consists of three base pairs, so an A, a U, a C, and a G, so three of those, and they're always complementary to each other, okay? So this chart here is the genetic code, all right? So this chart looks very confusing, okay, but it's not, I promise, all right? So this region over here to the left, this is representing the first, and this is for the codon. All right, so this is sometimes called the codon chart. So this region here to the left, this is the first letter of the codon. The one up at the top is the second letter. And then when you go into that box where the two overlap, you have various options for the third letter, okay? And you can see the third letters over here. So this top row is U, the second row is C, third is A, and then the last one is G, right? So say we wanted to find the amino acid that's coded for by the codon CGA or something like that, all right? So we find the first letter C, right? So we know we're in this row here. We said CG, go to this one here where the two meet. So we're in this box here, and CGA, all right? So CGA, all of these codons code for this amino acid, which is arginine, okay? So you can see here that sometimes a single, and in many cases, a single amino acid is encoded by multiple codons, all right? So we could have CGU, CGC, CGA, and CGG, and all of these code for arginine, all right? You could also have these down here, AGA and AGG. Those also code for arginine, right? So what this means is that there is some redundancy kind of built into this system, right? So you could have multiple codons, and they could all code for the same amino acid, right? So you can also think of this in the sense that it could be potentially a nice way to still get the same protein if for some reason you may accidentally have input the wrong base pair, right? So say in your when you're translating and you're laying down your mRNA base pairs and you meant to put down a CGA, but instead you put down a CGU or something like that. Well, we meant to put down a CGA, but instead we put down a CGU. Well, there's really no difference in the output, right? You still get arginine for the same case, right? But it's not always that case, right? So say we meant to put down CGA, but instead we put down CAA. Well, CAA is this other amino acid, right? So now we've completely created a different protein, different amino acid, and so the product of this protein synthesis in this case is going to be completely different, right? So sometimes it doesn't really matter in terms of whether or not you put down a wrong amino acid. Sometimes it matters very much so, all right? So here you can see again, our anticodons on our tRNA, our codon on our mRNA, and the two are always going to match up. So A goes with U goes with A, G goes with C, all right? So keep this in mind as we're looking at this, okay? Now, once we have our protein, we can then undergo a conformational change, okay? In other words, the protein is going to twist and be folded into various shapes, and we said earlier that the shape of the protein determines the function of the protein, right? If this process of transcription that we just, or of translation that we just talked about, if this happens on a free ribosome, then that protein is probably going to be used in the cell. And when I say free ribosome, what I mean is the ribosome is floating in the cytoplasm. It's not bound to an endoplasmic reticulum. Okay, and we'll talk about ERs later. If it is, if this process of translation is occurring on a bound ribosome, then that protein is probably going to be secreted out of the cell, or it's going to be incorporated into the cell's plasma membrane. Okay, so depending on where the translation takes place, you'll either have that protein go out of the cell, or it'll stay in the cell or be bound to the membrane. Okay, so there are four different structures, if you will, to proteins. All right, and these are called the primary, secondary, tertiary, and quaternary structures. Right. So the first one is the primary. This is simply and very straightforward the linear sequence of amino acids. Right. So during the process of translation that we just talked about, you have one amino acid. It binds to another amino acid, which binds to another, and to another, and to another, and so on and so forth. This string, if you will, think of almost like a pearl necklace, right? All the pearls are the amino acids, and they're just strung together in a straight line. That is an example of a primary structure, okay? A secondary structure is when you have interaction of those R groups, right? So those R groups that we said, that standard for like the rest of the amino acid, so the variable part. And when you have interaction between different R groups of different amino acids, you get a secondary structure. These are called an alpha helix, which in this second region, second section here, that's this top one. This is called an alpha helix. And this bottom one is called a beta pleated sheet, okay? A beta sheet or a beta pleated sheet, right? And those are examples of secondary structures. You can also have tertiary structures, right? Tertiary structures are generally the overall shape, the kind of final globular shape of a protein. So proteins are kind of globular. They're not always just flat and linear. And this has to do with various interactions between different amino acids. So hydrogen bonding, ionic bonding between different amino acids, and you get a more complex three-dimensional structure. So if you look here on this image to the left, you get something that's a little bit more globular in shape. Okay, it's kind of more rounded, if you will. All right. Now, you can get something called a quaternary structure. Right, and quaternary structures happen when you have two separate proteins, so two separate polypeptide chains, come together and make one functional protein. All right. So a good example of this is the protein hemoglobin, right? So hemoglobin has alpha parts, alpha subunits, and it also has beta subunits. So these are different proteins that are interacting together to form one functional protein. And in this case, hemoglobin is used to bind and transport oxygen and carbon dioxide because of the iron units that are embedded within this heme unit here. So it has globulin units, it has these heme units, and then within these heme units that you see on this image to the left, there's an iron atom, an iron molecule, and this is what actually binds the oxygen. Okay, you'll talk more about this hemoglobin in AMP2 a little bit. Okay, all right. So we're going to stop here with this part, and then we're going to pick up by talking about what happens to the protein after it's made, after it's folded. How do we get it to its ultimate destination, either in the cell or outside of the cell? And that will be the topic of the next lecture.